So now we're moving to our late afternoon session. And this one's on technology collaboration. So now we're moving to all the new things that are happening and how we're going to use some of these technologies. So we're going to have our next session is going to start with Liv Liberman from, she's the program manager for Revive and Restore, and then followed by Aaron Mueller from the Moat. So Liv, please join us.
All right, let me see if I can do this. I can. Wonderful. Hi, everyone. Good afternoon. I hope you're all feeling caffeinated and ready to go still. My name is Liv Liberman. I work for an organization called Revive and Restore. We're a US-based nonprofit that brings biotechnology to wildlife conservation. And I'll be talking to you all about biotechnology and wildlife conservation today. We are US-based. We have projects all over the world, but I am a proud Floridian, not born and raised, but I've been here for about a decade, and I absolutely call it my home. And I love this state very much. So I will do my best to bring in Florida examples as much as I can. There's quite a few in this presentation, but we do have projects all over the world. So I'll also be highlighting some work that's outside of Florida that can hopefully benefit what we're doing here in Florida as well. All right, so I'm really grateful to be here today at the CAC conference. This is my first one, and I'm especially delighted that the co-theme, in addition to climate change, is biodiversity, because these two topics, these two crises, are often seen as mutually exclusive or kind of not the same conversation. But to me and my colleagues, and hopefully to many of you, to me it's a Venn diagram where the biodiversity crisis and the climate crisis really overlap, and in some cases are kind of becoming just a circle. They're absolutely so intertwined with each other and really cannot be parsed or separated. And so many of the things that we traditionally think of as causing the biodiversity crisis, right? Habitat loss, disease, overexploitation, invasive species, all of those things I'm really grateful to my former speakers have been talked about here today and are very much happening here in Florida, right? But each and every one of those problems is exacerbated by climate change. Many people think of climate change kind of as its own stressor. If I'm a coral, I'm stressed by this heating problem, and that's separate from everything else. But no, really, climate change, ocean warming, land warming, all of it feeds back into each of these other problems. Your invasive species, their ranges expand, their competitive abilities in many cases expand because of climate change. Disease, many pathogens become more virulent under changing climates, especially under warming climates. Habitat loss, right? As the climate warms, that changes where different animals and different organisms can live. And so each of these sort of other stressors are not separate at all, but they're absolutely linked to and exacerbated by climate change. And that's why we should really think of these together when we think of biodiversity loss and climate change. And what this really leads to in many cases is this extinction vortex. So what is the underpinning of resilience, of a population being able to survive and thrive in this world? It's resilience, and resilience comes from diversity. That's true for us humans, and it's definitely true in nature. And diversity, if you really kind of look at what that is, it means genetic diversity. The very basic building blocks of life, cells and DNA, are really what make us look different from each other in this room, have different susceptibilities to different conditions. That's very much true for our wildlife as well. So populations rely on genetic diversity to have resilience, to be able to respond to stressors. And what we're seeing all over the world for lots of different reasons is this kind of extinction vortex where we're losing animals, we're losing diversity. And as populations get smaller, that genetic diversity, as you can see with kind of these colors changing, right? Each color is representing a different genetic individual that has different traits genetically. As populations shrink, so does the kind of the building blocks that they have to work with to respond when disasters happen, to evolve and respond to natural selection as well. And that becomes kind of a feedback loop, as we've heard from many of the other speakers here today. As we lose genetic diversity, that then makes us less resilient when more disturbances come up, and we're not able to adapt and eventually end up with very inbred populations that do not have kind of that basic currency with which to operate under natural selection. And what we're hoping to do, us in this room and all over the planet as conservationists and as people hoping to help our wildlife come back, is reverse this extinction vortex kind of by going in the opposite direction. If we're in this position now, right, where we have many, many populations of animals, organisms all over the world that have shrunk because we've shrunken their habitats, because they've had disease outbreaks, because invasive species are encroaching on them, because the climate is warming, et cetera, they now in many cases are inbred. They have very low genetic diversity. There are few individuals and thus very low genetic diversity. But if we're able to either bring new individuals into that population, or if we're able to increase that genetic diversity itself within that population, we can actually reverse this trend. The opposite of inbreeding is outbreeding, being able to increase that kind of genetic variation that's there. Higher diversity then means they can reproduce better. They can potentially grow that population and make it more resistant to new stressors that come out. And that leads us eventually to a more stable population. And so we're hoping to be able to do this. My organization, Revive and Restore, is all about this genetic rescue concept. How can we kind of harness the power of cells and DNA to increase the genetic diversity of our struggling wildlife and ecosystems out there to hopefully make them more resilient into the future? And we can really learn from many of these fields that are already out there, particularly medicine and agriculture, that are already harnessing the power of cells and DNA through a number of technologies that I'll talk to you about today to, understand what makes us human. The Human Genome Project, for instance, is a great example of that. We're able to cure and treat diseases today because we understand the cellular and genetic mechanisms behind them, and we can actually predict how those will happen. In some cases, we can even manipulate and change those outcomes for people to cure diseases that we could never cure before. And then similarly, we're able to feed, to some extent, this explosive population growth that we've seen on many of the graphs today. Of course, not perfectly, but kind of astonishingly, because of many of the advances in agriculture that in many cases come from really biotechnology. And so what if we borrowed this toolkit from biomedicine and from agriculture and brought it over into the realm of wildlife and endangered species, because they're also made up of the same exact building blocks, cells and DNA, and can probably be, understood and potentially manipulated in some of the same ways. And so Revive and Restore kind of came up with this concept of a genetic rescue toolkit. That's what you see up here. This is not exhaustive, but it's sort of how we think of the different tiers and levels of tools that can be leveraged to help with wildlife conservation. The kind of basic bottom building block there is just sequencing, genetic sequencing, understanding what is there in a population, and thus being able to understand better the adaptive potential of that population. How inbred is it? How resilient is it? How can we use that information to make better decisions? I'll be talking about these other tools throughout the rest of the talk, so I'm not going to give you too much of an introduction to them now. But let's start with that sequencing level that I mentioned, because it really is the foundation of everything else. So there's amazing technologies out there that allow us, the Human Genome Project is a great example, to understand really like what we're made up of on a molecular and cellular level. You can, we know the human genome, it's amazing. And that tells us all about who individuals are, how related to each other they are, where they come from, what their evolutionary history is in many cases. You can look at, in some cases, DNA material, if it's preserved well enough, from things like fossils, and understand the relationship between things that lived a long time ago and things that are living today and those changes. And really it tells us about how much currency there is within a certain population to kind of work with from a natural selection perspective. So I'll show you a few examples of how genetic sequencing has been incredibly helpful for wildlife conservation. One of our favorite examples, of course, is the genetic rescue of the Florida panther. Back in the 1990s, this population was incredibly inbred. There were very few individuals. They had very low genetic diversity, and they were just sort of in that extinction vortex that I showed you earlier. And really thoughtful and kind of forward-looking folks in Florida and elsewhere thought that it might make sense to translocate some of these panthers, the closely related, same species, but sort of a subspecies of panther from a population in Texas, to inject basically a new bunch of genetic diversity into this inbred population. And not that our panthers are out of trouble, they certainly are not, but they've really kind of reversed that trend and been able to get away from that incredibly inbred state that they were in back in the 90s. And studies continue to show that process did not kind of get rid of all of the important adaptive potential that they had from here in Florida. Sometimes you worry about outbreeding depression, where you would kind of get rid of all of the nice local adaptations that are good for a population when they're, adapted to a certain place. So it actually, that same population was able to keep all of the, adaptive potential from here in Florida, but they also got rid of that inbreeding problem. This homozygosity term means that basically they're way less inbred. They reduced homozygosity. They reduced that inbred nature of the Florida panther population. And so just sort of through genetic sequencing, we're able to track these trends. And translocations like this could be incredibly helpful for all kinds of other species that are becoming isolated, with populations that are becoming small and inbred in many cases. Corals is actually one of the places we're looking into starting to potentially do translocations to try to genetically rescue some of these small inbred populations. But Florida really stands up as the shining example of one of the first times that this was tried and really succeeded. And so I think we can all be really proud that, our state led the way with this particular type of intervention back in the 90s. And just to show another example, these guys are not from Florida. This is the sunflower sea star, beautiful, crazy, multi-armed sea star that's native to the west coast of the United States. And unfortunately, they have been absolutely decimated by sea star wasting disease that's killed, some estimates are 99% of the populations off the coast of California, Oregon, Washington, and up into Canada. And luckily, they were recently able to identify the pathogen for this disease. So that's a good start as far as being able to understand, how to maybe treat it. But importantly, of those 99% of sea stars that died, some survived. And why is that? Most likely, they have some sort of genetic variation in those animals that allowed them to potentially be more resistant to that disease. And so studying what it is about their genetic code that might have actually allowed them to survive that disease outbreak is really critical. So we and others have funded projects to look into the kind of genetic markers of disease resistance in these sunflower sea stars and been able to identify them, which are now being integrated into captive breeding populations, so that we're not only breeding back sea stars that we're going to put back out into the water that would, meet the same fate as all of the other sea stars and succumb to that disease, but hopefully so that they can go back out into the world and not succumb to that disease and be more resistant. So the power of genetic sequencing is able to just show us, what are those markers that underpin some of that resilience, and how can we harness that in our ongoing conservation programs like captive breeding, so that we're not just flying blind, so that we know what individuals we're putting out in the water, and thus how likely they are to probably succeed in that intervention. Okay, up next we have biobanking. So this is a topic that I believe my colleague Erin will also touch on in corals, but this is the process of preserving biological living material, whether that's cells, tissues, even full organs in some cases in humans, which is amazing. Scientists have developed methods to actually cryopreserve or freeze these living cells and tissues so that they can sort of be saved in the long term for long-term, almost indefinite storage, and can actually be revived or thawed in the future and continue to live. The cool thing about these cold storage methods is that basically at super, super low temperatures, like the temperature of liquid nitrogen, negative 196 degrees Celsius, I believe it is, all biological and chemical activity basically stops, and that includes biological and chemical activity that leads to death. And so you're just frozen, and you don't die, and you don't, degrade if the right methods are followed. It's not that simple. But it's really amazing because cells and DNA, importantly, genetic diversity, importantly, can be safeguarded and prevented from being lost forever. So this is a really, really important first safeguard against the loss of genetic diversity in species for whom we are losing individuals all the time. And because you can thaw them and they can come back to life-ish, and you can use them in the future, they're really important materials for future interventions, for conservation interventions, whether that's 2 weeks from now or 200 years from now. So there's some really amazing efforts out there to do biobanking for wildlife. The San Diego Frozen Zoo has been around now for 50 years, which is incredible. And they have, I don't even know the numbers, I really should, tens of thousands of frozen samples of all kinds of taxa around the world and are really leading the charge as far as biobanking. Similarly, the Smithsonian's National Marine, Marine, I always want to say because National Museum of Natural History has a biorepository with all kinds of samples, again, with cells, tissue samples, DNA, et cetera, of all kinds of organisms. And these are really important repositories. But the infrastructure, as you can imagine, for some of these things, you require space, you require the liquid nitrogen, the sort of dedicated long-term funding to be able to keep these systems up not only for years, but hopefully for centuries. And so these actually are quite few and far between. And one of the biggest bottlenecks to biobanking is really that capacity, that physical infrastructure, and the kind of long-term funding to be able to maintain some of these systems. So I just wanted to throw that out there for anyone who's interested in biobanking. But I do want to highlight that some really awesome Florida species are being biobanked. Revive and Restore has a project with the U.S. Fish and Wildlife Service called our Informed Biobanking Program, where we've developed all kinds of standardized protocols to biobank tissue samples and cell lines from U.S. endangered mammals and birds. And the Florida bonneted bat is this cute little guy, endemic to Florida, and is one of the species that has been biobanked, both as far as cell lines and tissue samples, and has also been created a reference genome as part of that. And I actually just learned from my colleague that there are several other Florida species that are involved in this project that I didn't know before I made these slides, otherwise I would have put photos on them. So let's see, part of that effort is also the key deer. These are our endemic species that we can feel very proud of. The key deer, the Florida scrub jay, the Key Largo wood rat, the Everglades snail kite, and the Anastasia Island beach mouse, and more. But those are just some of the ones that you may have heard of that are part of our Informed Biobanking project with the U.S. Fish and Wildlife Service, which is fantastic that those species are being saved and sampled, or at least were being saved and sampled by our federal government. And that work does continue and has not yet been jeopardized, so hopefully that will continue. All right, and then I also wanted to highlight, I'll again let Erin talk a lot more about biobanking in corals because it's really exciting and so important given how quickly we're losing our coral reefs. But we're also funding brand new technology in this space. For instance, the first attempt at cryopreserving and reviving entire coral fragments. So right now we're able to cryopreserve coral sperm and coral larvae, but that actually really limits the number of corals that we can work with because that limits us to only the species that we know the reproductive biology for so that we can access said sperm and larvae, which is actually a tiny, tiny fraction of all of the coral species that are out there. And as you'll probably hear from Erin a little bit, corals only spawn on a few nights a year. They're very specific, and so you really have to capture all of that material and biobank it or use it immediately. Otherwise, you have no chance at doing that biobanking. And by being able to cryopreserve just a fragment of a coral, which is a coral, you will be able to kind of open up how many different species you can biobank and also the different ways that you can use them, because it's not just a sperm, it's not just a larva. You can do a lot more with a coral fragment. So we're really excited about this technology. It was borrowed from human organ cryopreservation. So one of the scientists on this project is working in human medicine and using this same technology to cryopreserve, I don't know, kidneys or livers or something, some human organ. So that's super amazing, and it's a really direct example of how you can literally borrow technologies from these other fields that in a lot of cases have more funding than we do in conservation, and try to kind of adapt them for endangered species. All right, so next up we have cell culture. This is being able to understand that organisms, right, are made up of cells, and so in many cases being able to actually tease those cells out from the organism themselves and culture those cells long-term, separate from that organism, gives us something that we can study without having to go back to that organism, especially if it's an endangered species, and sample it over and over again. Especially in disease work, this is really important, because if you can study, the cells of I'm always going to come back to coral. I didn't say this at the beginning. I'm a marine biologist. I did my Ph.D. in coral reef conservation, so that's always what I'm going to come back to. But I'm going to leave most of the coral stuff to Erin. But if you have a coral, an endangered elkhorn coral, let's say, and I need to test, what is going on with it as far as its disease resistance. Can I, is this new treatment that I'm developing going to have a effect on it? I don't want to have to take a bunch of elkhorn corals that are already basically non-existent on our reefs to be able to test that. If I can test it on their cells, because we've optimized cell culture conditions to be able to keep sort of an infinite line going, that really opens up research and potential conservation interventions that we can test and eventually deploy in the real world with these real animals. And another really cool way that we can use cell culture is to develop microbial treatments, probiotics. So just like us, we have all kinds of little microbes in our body, in our guts, that help us, that are beneficial for us, that keep us going, and so do many, many, many animals and organisms out there. And it turns out that corals actually have quite a few beneficial microorganisms, bacteria, various kinds of microorganisms that help them out and can even help them against disease. So this, you've heard a few times about stony coral tissue loss disease today. This is one of our beautiful brain corals here in Florida, Diploria labyrinthiformis, or the grooved brain coral. That is very susceptible to stony coral tissue loss disease. And we actually don't even know the pathogen for this disease yet, so our options are relatively limited. But a really cool thing that one of our partners has done at Revive and Restore is actually develop a high throughput screening platform to be able to identify, isolate, and culture beneficial bacteria from healthy corals that actually have activity against and can protect corals against stony coral tissue loss disease. And so you'll see some of those bacteria being cultured there in a petri dish, and then actually a cocktail of those probiotics, we call them, being applied to a coral. And this is kind of how it works. They're able to take mucus from healthy corals, use basically a lot of the same testing mechanisms that we use, again, in clinical medicine and human medicine and drug discovery to test the isolates of these bacteria against stony coral tissue loss disease in a lab setting. Then they're able to do kind of a trial in a controlled setting against coral cells and against other cells to see what that activity would be. And then once those are deemed to be helpful against this disease and safe, they're able to actually test it on real corals. And right now, these probiotic cocktails are being applied both in the field and in land-based restoration with partners here in Florida. I think they were actually developed first here in Florida, which is exciting. And also in the Dominican Republic, in Colombia, and in Montserrat. So all over the Caribbean where this disease has just been ravaging our coral reefs. This can be delivered both as what you're seeing up there, which is literally going onto a reef. You find a coral that is, actively seeing lesions. That picture that I showed in the last slide, you can see that disease lesion going over the coral. And without intervention, the likely outcome is that coral would die. That lesion would keep going, taking the tissue off, and it would die. But with these probiotics, we found that you're able to basically put a glorified bag over that coral, weighted bag, and inject it with a cocktail of probiotics for 30 minutes or so. Take that bag off, and that coral is now able to survive that disease. The lesion stops progressing, and they heal, and it saves the rest of what's left of that colony. In addition, they're using this treatment as a kind of pre-treatment for coral fragments that are propagated on land. There's many, many land-based nurseries, including here in Florida. Moat has one, Coral Restoration Foundation. Various groups in Florida are propagating many, many corals on land, but then they have to outplant them, and you hope that the corals that you outplant onto the reef are not going to be susceptible to this disease. And so they're also using these probiotic treatments as a pre-treatment for some of the species that are most susceptible to this disease, and it's preventing them in many cases from getting that disease when they're being outplanted, which is really exciting. So that's some of the power of cell culture. And I want to kind of piggyback on this to talk about stem cells. Some of you may or may not be familiar with the concept of stem cells. We have them. It seems like most organisms, or at least most animals, have them. It is the cells in the body that are undifferentiated. We've got, I got hair cells, I got skin cells, I got heart cells, I got all kinds of cells that make up lots of different organs and things going on with me and with each of you. But there are actually cells in each of us, and they're most abundant, of course, when we're little embryos and we're turning into all kinds of different bits and parts. So there's cells that can actually differentiate into any kind of other cell in the body. They're undifferentiated and have that kind of potential to become anything. And that makes them incredibly powerful for research and potentially for therapeutic purposes. And in humans, these are already being harnessed every day in medicine for research and for therapeutic purposes. I already explained why having a bunch of cells that can act as human organs or as parts of an animal. It could be useful for disease research, for instance. That is absolutely happening in medicine all the time. Your stem cells can literally be taken from you, turned into various organs that are sort of like your fake organs, and then tested with various therapies out there in the medical space right now. And in addition, because they can differentiate into any different tissue type or cell type in the body, they can actually be used to potentially replace diseased or damaged cells or tissues if you're, suffering from a given condition. And so these uses are very much happening in humans today. And so why can't they happen for wildlife as well? They have the same building blocks as we do. And so, some progress has been made in this space where amazingly sperm and eggs have actually been derived from stem cells in mice. A sperm is a type of cell, an egg is a type of cell. And so theoretically, those can be derived from stem cells with the right conditions. And so a lot of this technology has tremendous potential for species that really need help with reproduction in particular. A species like a rhino that's down to the last few females, how are they even going to reproduce and produce new offspring? Well, maybe we could take a stem cell from that female rhino and reprogram it actually into a sperm cell to be able to create reproduction. This is something that's folks are working on it. We are not there yet at all in rhinos, but this kind of technology has huge potential for all kinds of species around the world that need more help with this. For instance, we've been successful. I mean as a field, not specifically my organization, but we are funding this work in the gorilla, which is cool. We have been successful at deriving induced pluripotent stem cells, they're called, just basically stem cells that can differentiate into all kinds of different organs and different cell types in the gorilla. And my organization has a brand new program, the Applied Stem Cell Conservation Program, to specifically fund this type of work in all kinds of endangered species. Can we figure out how to take a cell, a somatic cell too, like literally a hair sample, it's not invasive, from any of these endangered species and reprogram that into a sperm cell, an egg cell, a tissue type that we need to study for disease. These are two of the species that we just started new projects to work on. The Ethiopian wolf, which is the most endangered carnivore in Africa, and also the Burmese star tortoise that is struggling, as many reptiles are, from temperature-induced sex determination challenges where the temperatures are really pushing them in the direction of producing really way more one sex than another. And so we're hopeful that maybe some of this stem cell technology might be helpful in tackling some of those challenges that they're facing. So we're really excited about those projects. They're brand new, but we're very hopeful about them. And then in corals too, corals have stem cells, and we're funding some of the only work being done in coral stem cells in the world. So this awesome team, Dr. Nikki Traylor-Knowles, is based at the University of Miami, so right here in Florida. I'm going to make as many Florida connections as I can. And her collaborator, Benjamin Rosenthal at Ben Gurion University in Israel, were the first to derive stem cells from corals, and now they are working on culturing those stem cells and have kind of mastered techniques for transplanting stem cells between individual corals. And so now they're testing this kind of as the basis of a genetic therapy. Can we take stem cells from a coral that we find to be more thermally tolerant, transplant them into a coral that's more thermally sensitive, and does that actually kind of vector that heat tolerance within the same generation of the coral without having to breed them, without having to wait and actually vector them? And so far, they're finding that these cells are really being accepted in many cases by the donor corals. They're not seeing really any rejection on the cellular level, which is amazing because that's definitely a problem with transplants of any kind in human medicine too, right? We're really worried when you get an organ transplant, is your body going to take that up? And so far, we're finding that corals seem to be really receptive to receiving cells from other corals, which is great and might enable this technology. So we're really excited to see where this continues to go. It could have many applications outside of heat tolerance as well, but we think that the heat angle is a really exciting one, and that's the one that we're looking into first. Oh, and that's kind of how that works. Sorry, I just explained that, but taking cells from a healthy and what we think is more resilient donor coral, and then transplanting it into a more sensitive coral to hopefully vector that heat tolerance. Okay, now on to advanced reproductive technologies, and I'm going to show you this through the story of one of our favorite little species, unfortunately not a Florida native, but I think you'll like them anyway, the black-footed ferret. This, at a time a few decades ago, was North America's most endangered mammal. Thankfully, it is not anymore. It used to be ubiquitous all over kind of the Midwest and Rocky Mountain West, from Canada down to Mexico, all on the prairies and in the plains. And this poor little guy, mostly due to habitat loss and changes in the ecosystem, of course, that were all human caused. We were hunting, I believe, the prairie dogs, which is part of their food. Anyway, they got down to 7 individuals back in the 1980s, which is not a population that can survive. Talk about an extinction vortex. And as you can imagine, 7 individuals, that's going to be very, very inbred, even if you're able to recover from there. And amazingly, the U.S. Fish and Wildlife Service was able to actually create a program at the time, 40-ish years ago, to say, oh my goodness, we're down to 7 animals, we got to do something about this. They created a captive breeding program and succeeded in getting us up to somewhere around 300 ferret individuals today, which is a great, bounce back. But remember, they all came from 7 individuals. So that population is really inbred. Their genetic diversity is really low. And it's really been starting to show. They're particularly susceptible to a disease called plague, which I will come back to later because we have another biotechnology solution to try to deal with that. But really the underlying problem is they're not very genetically diverse. How do we do something about that? So the U.S. Fish and Wildlife Service came to Revive and Restore, let's call it a decade ago, and said, can you help us with this? What can we do about this? Is there a biotechnology solution? And we were able to figure out that there was actually a frozen, cryopreserved, biobanked, yay biobanking, we love biobanking, sample of an 8th ferret individual, so totally different from any of the 7 individuals that had been remaining alive, that the whole breeding program came from. A ferret that died 40 years ago, but somebody had the foresight to cryopreserve some really high quality cells from. We were able to identify that specimen existed and was high quality enough that we could actually use cloning to restore that genetic diversity back into the ferret population. That works by basically taking the nucleus, the cell, the nucleus, the place where all of the DNA and information is, from that biobanked cell, from that 8th ferret individual, and actually transplanting it into a domestic ferret cell. So very closely related to the black-footed ferret is the domestic ferret. So you take out the nucleus from the domestic ferret cell, you're actually able to insert the nucleus from the cryopreserved black-footed ferret cell, and that then develops into an embryo that is a black-footed ferret because it has all the genetic information, the nucleus, that's your entire genetic code, from that black-footed ferret. That embryo is then transplanted into a surrogate mother that's a domestic ferret, and the domestic ferret gives birth to a black-footed ferret baby. And so in 2020, Elizabeth Ann was born, the first cloned North American endangered species. She's so cute. We love her. And since then, 2 more clones have also been born. And what's really exciting is that all of these clones have now also reproduced naturally with non-cloned animals. So that means we are literally seeing the genetics from that 8th ferret individual go back into the population that was really, really inbred. And various genetic studies have shown us that the allelic diversity, the diversity that was there in that 8th ferret individual is maybe up to 3 times higher than any of the individuals that came from that original 7. And so just by restoring kind of that historical genetic diversity that would have existed before the population got so bottlenecked back into this population has really given a lot of hope for this species. And we were able to do the same thing with the Przewalski's horse, which is the only true wild horse species that are native to China and Mongolia and Ukraine and Russia, kind of the Russian steppe area. They were similarly down to a dozen or so individuals a few decades ago, and now 2 clones, Kurt and Ollie, have been born through cloning. And so now we're waiting for them to mate and bring their genetics back into the population as well. So this is a tool that has not, to my knowledge, been used on a Florida species, but we have many, many small populations of inbred species, and this is a tool that could potentially be used if we have really good biobanked samples to work from. All right, and then my final kind of category here, I'm going to talk about genetic engineering and gene editing. And so this is CRISPR. You've probably heard of this. It is a kind of on the more risky end of tools, but one that we're using all the time, again, in medicine. And it's really allowing us to make very precision, targeted edits into a genome to specifically target a certain section, a certain gene, a certain nucleotide in some cases to tweak little things about the genetic code with the idea of potentially increasing resistance, resilience, genetic diversity. Depends on what the target is, but I'll show you some examples of where this has been really powerful or could be really powerful. Actually, as Floridians, here's an example that comes from human health, not from wildlife, but it's just to show you that this technology has been used out in the world already with the goal, at least, of benefiting humanity. So, mosquitoes, as we all know, are disease-causing, are hugely problematic around the world. And here in Florida, actually, a company called Oxitec created a strain of genetically engineered mosquitoes that had a lethal transgene, a lethal edit, basically, that the males carry that actually is only expressed in females. So the males carry it. They're able to release a bunch of males that have this edit in their genome. They're flying around. They're doing fine. They mate with females, and any of those female offspring immediately die. Female offspring cannot survive. And so that already takes out kind of half of the population, at least, of that of those offspring. And that's important because the female mosquitoes are the only ones that bite and spread disease. Males do not bite and spread disease. And so by releasing more males out there, you're not, releasing extra mosquitoes that will bite and spread disease. And so in theory, with enough of those, that would kind of self-limit the population away from being able to reproduce at all. In 2022, they actually released a bunch of these in the Florida Keys after a very lengthy process of, community engagement and government approvals and all kinds of things in the Keys. And they at least claimed that they were able to see some of the effects that they were looking for, but it was too small of a, pilot study to be able to wipe out the whole mosquito kind of population. But just wanted to share that here in Florida, this has happened for the purpose of human health. And although it hasn't happened, there's a lot of talk of using those same kinds of either lethal or, bad for the species types of edits and gene editing to potentially control invasive species. Here in Florida, we've got a lot of those. The Burmese python, the lionfish is a problem in our coral reef domain, iguanas, the Cuban tree frog, all kinds of different things. And many projects have been proposed. Very few are underway, but to be able to kind of try to create a lethal, very targeted, so it's only species specific type of, intervention for an endangered species, or excuse me, not endangered species, invasive species that would not harm any other species. Again, these have not happened, but these are conversations that are kind of happening, and researchers are definitely interested in doing this work. It's just not quite underway yet, but it could be used. And then as an example of where gene editing and genetic engineering could be really helpful for endangered species, I'm going to come back to the black-footed ferret. So I mentioned that they're really susceptible to this disease called plague. Amazingly, their domestic ferret relatives are not susceptible to this. So we've been able to kind of study the domestic ferret and figure out what some of that basis of resistance is. And this has really caused a problem for the reintroduction of these ferrets out into the wild. Many of the places that they've sort of been released are now experiencing disease outbreaks and threatening this restoration activity. Fortunately, a vaccine has been developed for this disease, but it's injectable. So that means you got to, catch the ferret, inject the vaccine, and unfortunately, it also needs a booster for it to be effective a month later. So you got to catch the ferret again a month later. As you can tell, that's not scalable. It's not really feasible for a wild population. And so one of the things that we've developed and are working on in conjunction with our partners at U.S. Fish and Wildlife and the Morris Animal Foundation is a genetic vaccine. It's basically a circuit type of technology where you can put in a synthesized genetic sequence that turns on only in specific tissues at a specific age and basically pumps out the antigen, the protein that has to do with that disease, to train the ferrets, similar to the vaccines that we get, right, to train the ferret's immune system to recognize that disease and attack it. And so we're working on this project to develop a genetic vaccine for the black-footed ferret. The wonderful thing about this is that it's in the genetic code, it would be heritable. So once you put it into, a few ferrets, they then reproduce, and all of their offspring also automatically have that genetic vaccine and are not susceptible to that disease. So this is still in development. It's not out there in the world yet, but it's a really powerful technology, we think, that could be used in many, many other wildlife diseases if it's successful here. And you put that into the ferret cells, very similar to the cloning sequence, where the black-footed ferret cells with the genetic edits would then be put into the cell of a domestic ferret that creates an embryo that has, again, the black-footed ferret DNA in it, but it's kind of the machinery, if you will, of the domestic ferret embryo and developing embryo that's then put into a surrogate mom and born as a black-footed ferret that has that genetic vaccine in its genetic code. So really cool stuff. So we're really hopeful that this will work for us. And then one more quick example of genetic engineering. We also are funding a project led by Dr. Phil Clevis at the Carnegie Institution to investigate whether CRISPR might be helpful for increasing heat tolerance in corals. There are many, many genes out there that are potentially implicated in coral heat tolerance, and they've developed a high throughput screening platform to be able to actually identify specifically which genes are implicated in heat tolerance and be able to go in and potentially edit those. And here's an example, for instance, of a gene that was involved in symbiosis establishment. That symbiosis is critical to bleaching and heat tolerance, and thus by knocking the gene out, that coral doesn't even establish symbiosis, as you can see there. So we're really excited about where this could go as potentially a tool to help with coral heat tolerance in the future. All right, and then really, really quickly, this can also really help and come back with, am I going over time? Wow, I'm surprised by that, actually. Sorry, I'm almost done, I promise. Really quickly, these tools can specifically be used for species that are helpful actually for climate mitigation. There are many creatures out there, from the tiniest little microbes to vast mangrove forests, that are naturally carbon sequesterers, methane sequesterers in some instances, and really all of that capability comes down to what they're evolved to do and what their cells and DNA can do. And so many of these tools can probably be leveraged to potentially increase their climate mitigation capabilities. So I am going to stop it there and say, thank you so much for listening. I just want to make it clear that these solutions, while they're incredible, they're all meant to be complements to conventional and traditional environmental restoration approaches. They are not a replacement for habitat protection, for stopping of overfishing, for all of the things that we're doing already. They're a complement to all of our more traditional approaches and should not be seen in a vacuum. And hopefully, with all of those together, we can really help some of these species and ecosystems. Thank you. Sorry. That's good.
All right, any questions for Liv?
Okay.
Right here.
I just want to say, after that, now you know why I am so optimistic about the future. Wow, that was amazing. Thank you.
Yeah, of course. Yeah.
Definitely wow. So I work alongside Dr. Lacavera. So you can imagine the general public, when they hear of technology like this, think we're going to turn prehistoric mosquitoes into T-Rexes chasing Bob down the roads of Sarasota.
Before, yes.
So we get this question a lot that questions the ethics of using what are perceived unnatural processes to impact the natural world. How do you respond to people who say that?
Thank you so much. That is a great question, and one that we get all the time, and one that I would have said if I had not already gone over time, so I apologize. My argument to that, I think there's many, many ways to address it. My argument to that is, as much as we would like to think that nature is natural, nature's not natural anymore. We have had such an impact on nature all over the world. There is no ecosystem that is pristine. There is no species that has not been touched by our influence. We have been playing God for the last certainly 150 years. I would argue certainly a lot longer than that. And so if we can use a lot of that amazing ingenuity that has led us to this place where we've, created amazing technologies that have kind of hurt the world so much, what if we could turn that ingenuity around and instead harness it for good with, instead of thinking about what are the unintended consequences of some of these technologies, what could go wrong? If we're doing it for the purpose of conservation, not for the purpose of profit, which is what we've done in many cases where many of these invasive species have come from, if we can turn this ingenuity for good and for, understanding that we're trying to help these ecosystems and keep them from going away, I think it's our moral obligation, to be quite honest with you, because we've caused all of this damage in the first place. And I would also weigh the risks against the cost of inaction. We know the cost of inaction based on all of the horrible graphs we've seen today. If we don't, not for every single species, but for quite a few of them out there, if we don't act with enough urgency, they're not going to be here. And that's going to hurt them, but it's also going to hurt us.
All right, Liv, last question right here. Pam?
Oh, great. Thanks. Wonderful talk. I really appreciated that. And I mainly just have a comment and an offer. So at the Florida Museum, we also have a genetic resources repository, and we have about 150,000 species that are, or samples that are in that repository. And we would be happy to share them. That's part of what we do. And if anybody here is interested in contributing to further developments along these lines, we'd be happy to work with you as well. So thanks.
Thank you, Pam.
Thought she was going to make you a job offer on this spot there.
Thank you.